Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notification. In this video, I am going to explain one of the topic in transport in plants. It is phloem transport, flow from source to shrink along with Gridling experiment. In plant body, the long distance movement of organic substances or food material, it always occurs from source to shrink present in plant body. Usually the source, it can be understood by some of the part which can synthesize food materials are called source. Example, green color leaves and young stem have a capability for the synthesis of food material. And some of the part that need or store food material are called shrink. Example, the growing pods present at the apical portion of the plant body along with storage part act as a shrink. The source and shrink, it may be reversible depending upon season or plant need. Generally, the sugars are stored in root and mobilized to become source of food in early spring season when the bud of the tree act as a shrink and required energy for their growth and development of photosynthetic apertures. In this, at the earlier season, when the leaves are well developed, it acts as a source and root acts as a shrink. But depending upon season, at the early spring season, all leaves are fall down in a tree. At this time, the stored food materials present in root, it acts as a source and newly developed leaf and buds acts as a shrink, so that the relation between source and shrink, it will be changed depending upon season and needs. The source shrink relationship is variable, especially the direction of movement of phloem can be upward or downward, so that it is referred to as bidirectional. Along with phloem, xylem, it helps in the movement of water, and this water movement always unidirectional towards root to stem in upward, and one way flow of water take place due to transpiration by leaves. The food materials in phloem sap can be transported in any required direction so long as there is a source of sugars and shrink able to use or store or remove sugars, because the source continuously synthesize food material, and where it is utilized, that can utilize or remove sugars, that leads to transport of organic food materials through phloem tissue in bidirectional take place. The phloem sap, it consists of water and mainly sucrose along with other sugars, hormones and amino acids are present in phloem sap. It helps to transport or translocate through phloem tissue. To explain this flow from source to shrink, one of the hypotheses called the pressure flow or mass flow hypothesis, it is proposed by some of the scientists, and it is the hypothesis accepted mechanism used to translocation of sugars from source to shrink is called pressure flow hypothesis. This pressure flow hypothesis, it was proposed by E. Munch and it is elaborated by Grafts. So during this translocation, glucose which is prepared in source, especially leaf, by photosynthesis, this glucose first it converts into sucrose, and In plant body, the translocation of food materials always takes place in sucrose form. It is a disaccharide. According to this mass flow or pressure flow hypothesis, the organic substances in the form of sucrose, it always from flow in solution from sieve elements like phloem due to development of an osmotically generated pressure gradient in between source and shrink ends. Mesophyll cells are the only few cells away from sieve tube elements and companion cells, and the food materials which are synthesized in mesophyll cell, it can act as a sucrose, and in this diagram it is denoted with A, and the utilized or shrink portion is denoted with B. Due to creating a gradient of pressure, it can be transported from A to B like source to shrink according to mass flow or pressure flow hypothesis. During this translocation, the sucrose which is synthesized in green leaves or source, it moves into companion cells first. After this, these sucrose then it can be transferred into living phloem sieve tube cells by active transportation method. Because in the phloem sap already sucrose is present, so that due to concentration gradient it is not transported into phloem sap, but against concentration gradient sucrose is loaded into sieve tube cells by active transport method by utilizing ATP energy. So the process of loading at source, it leads to formation of hypertonic condition in phloem cells. Due to this hypertonic condition, the water which is moving through xylem, because of concentration gradient, it can transport from high water potential to low water potential from xylem to phloem tissue by osmosis process. Due to entry of water into phloem sap, it build up pressure, and this pressure which is created in phloem sap will move the area of low pressure where it is present. At a shrink region, osmotic pressure it must be reduced. So it is the source having high pressure, it is the shrink having low pressure. So again active transport is needed to move sucrose from phloem sap to outside of the adjacent cells, and by utilizing ATP energy, the removed sucrose at shrink it will be converted into either energy or starch or cellulose, but it never came back into phloem sap at shrink to maintain low pressure. As sugars are removed, that leads to osmotic pressure decrease at a shrink. When sucrose is removed, automatically in phloem sap, the osmotic pressure is decreased due to removal of water from from phloem sap to xylem cells, that creates low pressure. Usually, due to loading of sucrose at source into phloem sap and due to entry of water from xylem at source in phloem sap, high pressure or osmotic pressure is created due to removal of sucrose at shrink, and due to removal of water from phloem sap to xylem, low osmotic pressure is maintained at shrink. Due to this pressure gradient, the movement of sugar, more specifically sucrose, through phloem, it begins at source, and sugars are loaded actively into sieve tube elements, and loading of phloem set water potential gradient in phloem tissue. The phloem tissue is composed of sieve tube cells. These are arranged one above the other in the form of a long column with minute holes in their end walls called sieve plate, and these sieve pores are helpful for the transportation of sucrose through it. The cytoplasmic strand present in between two sieve tubes through holes or sieve plate, they form continuously in the form of filaments. When hydrostatic pressure in the phloem tube is increased at a source, that can sap moves through the phloem tissue. At the sink, the incoming sugars from source are actively transported to outside of the cell, and due to removal of carbohydrates, the pressure will decrease at shrink. So the loss of a solute at shrink, it produces high water potential in phloem sap, so that water again removed from phloem sap and returned eventually into xylem to create low pressure at shrink. That is about the translocation of food materials or organic substances from source to shrink according to mass flow or pressure flow hypothesis. During this translocation, phloem tissue it play an important role, and this phloem tissue it is useful for the translocation of organic substances that can be demonstrated with the help of an experiment is called Gridling experiment. In this experiment, it is a simple experiment called Gridling, and it was used to identify the tissues through which food is transported. For this, on the trunk of a tree, a ring of bark up to a depth of the phloem layer can be removed carefully. This narrow ring of bark it consists of phloem, cortex, and cork in its structure. After a few weeks, the bark above the ring on the stem it will be swell. The leaves do not wilt, but growth below the ring was reduced after removing this ring after few weeks. So due to absence of downward movement of food, the portion of the bark above the ring on stem become swollen after few weeks. This swelling of bark above the ring, it was due to accumulation of nutrients in that region as translocation of sugars and other minerals down. The plant was stopped, whereas the upward movement of water is not affected, because when we remove the ring, the xylem can transport water to upward direction, but the movement of food materials towards downside is affected, so that the lower portion it will be affected in their growth. This experiment finally it proves that the phloem is the tissue responsible for translocation of food material in one direction towards root, that is Gridling experiment. So it is a concept related to transport in plants and flow from source to shrink, and phloem tissue is responsible for transportation of organic food substances. Thanks for watching.